So, firstly, thank you very much for inviting me to speak today. I'm going to be speaking about monoclonal antibodies and more specifically a lateral flow device for invasive pulmonary aspergillosis diagnosis. I come to you as both an academic at the University of Exeter, so I will be talking about some pure science actually, as opposed to just sort of the more clinical applied stuff. But also as a director of a university spin-out company, Diagnostics. I get asked all the time, why is your company called Diagnostics? It's basically because Isca is the Roman name for Exeter, because it's a Roman city, walled city, okay, in the southwest of the UK. And if you go around Exeter, everything is called Isca. So Isca fish and chips, Isca MOT. So we decided not to try and be flash and call it something really clever, we just called it Diagnostics. And that little thing's supposed to be, Isca refers to the River Exe that still runs through Exeter. Okay. So, yeah, I'm talking about monoclonal antibodies. So what I actually did is I put together a list of monoclonal antibodies that I've generated during my lifetime, okay? And most of them are against molds, because that's really where my background is. It's not really yeast, it's molds. But, so there's a whole range of different monoclonals that I've raised that are highly specific for all these different well-established species, for example, Aspergillus, but also for what are known as emerging molds. Some would argue they've already merged, okay, because they have been around for quite some time, but they're just not seen in the kind of numbers that you see Aspergillus. So you've got Pseudoleishmania Scedosporium complex, Scedosporium prolificans, Fusarium specific monoclonal antibody, Trichosporon, Trichoderma, not really a human pathogen, although there are reports of it in dialysis patients, etc. Geoticum, and more recently, in fact, in the last few months, developed monoclonal specific to Candida albicans. Okay, so and that's actually funded by OLM Diagnostics, who are here today. And they're the sister company to Diagnostics. So they're the company that distribute and market and sell the Aspergillus lateral flow device. And shortly, hopefully, a lateral flow device for Candida albicans. Okay, so the one I'm actually going to concentrate on today is Aspergillus. And what you'll see actually from the diagnostic antigen column here is that most of these antigens are pretty uncharacterized, because actually when you raise monoclonal antibodies to fungal pathogens in mice, which is the standard species for making hybridomas, fungi are unique, perhaps, in that they elicit a very strong immune response against carbohydrate. And unless you've got multiple degrees in glycobiology, the likes of me are never going to be able to work out what they're recognizing. So they're recognizing carbohydrate epitopes on very large molecular weight glycoproteins. The exception here actually is this monoclonal antibody that's specific to Scedosporium prolificans, and actually it recognizes a protein as opposed to a carbohydrate. And so you can do some quite fancy stuff as an academic in a molecular genetics lab, which is where I'm based. And basically this is the monoclonal antibody specific to Scedosporium prolificans. So here what I've done is I've used a two-dimensional gel electrophoresis to separate all of the proteome within the fungus, and then done a Western blot on that 2D gel. And you can see that the monoclonal antibody is picking up very specific spots within that proteome. And if you go in and use mass spec, you can determine what that antigen is. And you can now sequence a 40 megabase fungal genome. We do it regularly now in Exeter within a week, and assemble the entire genome within a week using Illumina sequencing. So we have the genomes sequences to all of these pathogens, and basically we identified that as a tetrahydroxynaphthalene reductase, which is a protein involved in melanization of the fungus. So it's a dermatiaceous fungus, produces loads of dark pigment. And so what we did is we went and used homologous recombination to target the gene encoding the protein. And you can see on this one-dimensional Western blot, we have mutants here where we've knocked the gene out and we've lost the protein. So it's possible with proteins to actually identify what they are. And this is what happens to the mutants. So you knock that gene out, it's no longer able to produce the black menaline So this is a spore suspension of the wild-type strain, and these are the mutants, and this is the colours you end up with. What we've then gone on to do is knock out other genes that are involved in that pathway, and this is a skysalone dehydratase, polyketide synthase, more enzymes involved in that biosynthetic process. And there's a reason for doing this, because this albino mutant has no melanin, and melanin is known to be important to the virulence of fungi in humans. So we're interested to see what effect this is having on virulence and also on susceptibility to antifungal drugs. Okay, so that's the academic part, but really most of these monoclonal antibodies are pretty uncharacterized. We know that the Aspergillus antibody that's in the lateral flow device recognizes mannoproteins. Okay? So it's similar to galactomannan, but it is not galactomannan. Okay, so really why am I talking about invasive pulmonary aspergillosis and its diagnosis? And really because it's the most important mold species as a pathogen in immunocompromised humans, particularly patients with hematological malignancies and solid organ and bone marrow transplants. So it's an obvious first one to go for in terms of trying to develop a new diagnostic. So of course Aspergillus causes all sorts of different pulmonary diseases, aspergilloma, chronic, it's involved also in COPD, and I'll come on to that at the end of the talk. I think that might relate to someone's question. Okay, but really what we're interested in is this detection, where it's actually woken up and the spores have started to germinate and started to break out through the lung tissue. So clearly, if you're going to use diagnosis, you've got to try and do it early, as early as possible. And really, what you need to do is try and differentiate between where the lung is just being exposed to spores through normal daily exposure to the spores. We're sitting in this beautiful old building, and we're probably breathing in quite a number of Aspergillus fumigatus spores. Okay? It's always stated that at least every day, each individual inhales about 200 spores of Aspergillus fumigatus. I have no idea where that figure comes from, okay, but it's always quoted. But you will be breathing in quite a number of fungal spores, not just Aspergillus. So in a neutropenic patient, of course, you've lost your frontline effector cells within the lung that are going to clear spores that have been inhaled. And in the absence of those frontline effector cells of innate immunity, the spores will start to swell, and then they will germinate into hyphae, and then of course you lead on to your invasive pulmonary disease. So really you want to try and detect this early stage where the conidia are becoming active and are starting to grow into the active hyphal phase. So as we've already heard, there's no real gold standard for any invasive fungal disease. Okay? So you're relying on a whole load of different data coming from different sources and trying to put the picture together to try and best guess what's happening in these patients. So you're relying on the patient history, radiology, serological markers like galactomannan, beta-glucan. I've put molecular biology in brackets because I don't know whether Peter's fallen asleep. No, he hasn't. Is it going to be included in the next EO-RTC criteria, PCR? Okay. So at present, PCR is not a diagnostic, acceptable diagnostic assay for diagnosis of invasive fungal diseases. But of course, a lot of hospitals use it because it's a very powerful technique. Okay, and also histopathology and isolation of the pathogen from a biopsy sample. And of course, as we've already heard, it has to conform to the ORTC MSG criteria for diagnosis of possible, probable, proven infection. And clearly, the longer you leave diagnosis, the worse it becomes for the patient. So there's loads of different markers out there, and actually not just galactomannan and beta-glucan. There's a whole range of different PCR or nucleic acid-based techniques that are used for identification. Siderophores that are produced by the fungus. I understand this whole area has now been dropped. It's no longer going to be pursued, apparently. But then there's also metabolites like glyotoxin that can be detected using different analytical techniques. And pleasingly for me, the lateral flow device actually made it into this very prestigious review article. Okay, so it started to get recognition, but basically at present for invasive aspergillosis, you're really relying on diagnosis using galactomannan within serum and bowel fluid. Okay, so, but there really is plenty of scope for looking for alternative biomarkers for all of these diseases. There's many, many extracellular glycoproteins produced by fungi during active growth. It's just a case of finding the one that's most suitable for diagnostics. So you really want all, circulating antigens, things that are being produced by the developing fungus that go into the bloodstream and circulate so you can detect them within serum or indeed within bowel fluids. And the most appropriate really at the end of the day are clearly extracellular and are produced in large amounts constitutively. So they're only on when the fungus is actively growing and it's producing it all the time in large amounts. Okay, and really your diagnostics should be able to discriminate between active growth and sort of quiescent spores that are just lodged in the lungs and not actually becoming invasive. So I set out some time ago to develop alternative diagnostics for invasive pulmonary aspergillosis and screened many hybridoma cell lines, came up with a single cell line producing a useful monoclonal antibody, JF5, that's an IgG3, and what's great is it recognises an extracellular constitutive mannoprotein antigen. Okay, Aspergillus produces buckets of mannoprotein during active growth. There's a whole load of family, there's a huge family of mannoproteins. It's a superfamily, it's not just galactomannan. Okay, so it's a great target because it's produced in huge amounts. And the antigen is secreted during active growth phase. It's not produced during in dead or dormant spores. So what I did is I took that and stuck it into a lateral flow device in an attempt to try and get the diagnostics out of a crusty academic's lab and actually out into the clinic where it might actually be useful rather than just festering in a fridge. Okay, so little bit more information about the actual antigen itself. At the top here we have different developmental phases of the pathogen. Inactive dormant spore here, a spore that's starting to swell and then going through polar growth and producing a germ tube. And it's really at this kind of stage you're starting to get potential infection within the lung tissue. So the antigen, as you can see, this is immunofluorescence, is produced during active growth. So there's no detection of the antigen with the monoclonal antibody in the dormant spore. It's only where it starts to swell and go into the active growth phase that you get antigen. And this is immunofluorescence of an invasive hypha, and you can see the massive amounts of the mannoprotein that it's targeting being produced by the actively growing hyphae. So it's a really good diagnostic target for specifically detecting activity of the fungus. So, in terms of specificity, it's highly specific, recognises Aspergillus species, not just fumigatus. But all of these monoclonal antibodies are specific, so you can differentiate the different pathogens that you might come across in the clinical setting. So here we have Aspergillus fumigatus. I think batteries have run out. Or I've broken it. Oh no, it's back. Okay, so this is a hypha of Aspergillus fumigatus. This is Scedosporium apiospermum, one of these emerging molds. And this is a monoclonal against Scedosporium. You can see it's just picking up its own target species. And likewise here, the antibody that's specific to Aspergillus is just picking up Aspergillus. It's not cross-reacting, okay? So we can get a good handle on these different pathogens. And there are cases, in fact, in the literature of people having more than just one pathogen within the lung tissue. So there was a cystic fibrosis patient who died from multiple infections from yeast and also Scedosporium and Aspergillus fumigatus at the same time. So in terms of lateral flow technology, works on exactly the same principle as a pregnancy test. Okay, it's not novel technology, it's been around for a long, long time. Great thing for us is the patent ran out in 2008, which means that we can use it. So what we have here is actually I've got one in my pocket. So this is basically it. That's the diagnostic test, okay? And within that test, you have a strip nitrocellulose strip that has a release pad at this window here where you add your 100 microliters of serum sample or BAL fluid. And as the fluid goes in, if it contains the target mannoprotein antigen, it's picked up by the JF5 antibody that has 40 nanometer gold particles attached to it. Then the whole complex moves along by capillary action, and at a test line, you have the same monoclonal antibody that sits there, waits for the antigen to pass over. Okay, so your antigen... It's gone. Okay. So your antigen is picked up, attaches to the antibody, flows along, and where the antigen then meets the same antibody, it forms a sandwich. Okay, so you get a coloured line in the test line. You also have an internal control as a validation to show that the thing has worked. Okay, so where you have two lines at the bottom there, that's a positive test. Top, that's a negative test. So just the control line has come up. Okay.
Positive, control, negative.
Okay. Thank you very much. Can you come to all of my talk? Okay. So it's very, very quick. We're actually now saying that you read at 5 minutes. So 5 minutes after that application, you read the test. Okay. I've got 5 minutes. I've only got 5 minutes. Is that it? Are you going to tell me to... Oh, okay. I'll stop wittering. Okay. Okay, so in terms of specificity, highly specific, only picks up Aspergillus species, in this case Aspergillus fumigatus. It doesn't cross-react with any of the other sort of molds and yeast that you're likely to find in the clinical setting. So in terms of clinical trials, we've done about 8 now around the world, most of them in mainland Europe, particularly Austria and Germany. And I'll just summarise 3 of those where we've kind of increased the number of sites that are doing the test at the same time. So the first one was a retrospective single-centre study in Austria. And by retrospective, I mean it was based on frozen bowel samples. So these are banked samples. And we had 37 patients. The top 10 there were double lung transplants, kidney transplants, or just lung transplants. And what we did is we tested the LFD against galactomannan. And we got very good concordance between the different markers. Okay, so where galactomannan was positive, we also got LFD positive. We had a mucor species in there, Lichthemia, Candida albicans, both negative. What we found was, though, that the LFD cross-reacted with, and we know this anyway from analytical work, it cross-reacts with Penicillium species. Okay. We also got a glabrata positive there. We don't know why, because actually when you test glabrata, it's negative. Okay, but we got a weak positive there. Okay, so good concordance, and in fact, when you did all the statistical analysis, we had 100% negative predictive value for IPA in these patients. Okay, and then we moved on to a 2-centre study, again retrospective, using BAL samples, and I think it's fairly accepted now that when you use bowel samples and galactomannan, the ODI for the threshold for detection should be about 1. Okay, people might disagree with that, but I think it's generally accepted now. So what we do is compare the result for galactomannan, positive predictive value, negative predictive value, against the lateral flow device, and you can see that we've got again good negative predictive value in this patient group. And this was in hematological malignancy and various other patients. Okay, then we went on to, we've done a semi-prospective 3-centre study. Okay, so these are 3 centres in Austria, and this was, when I say semi-prospective, majority of the samples were tested as they came out of the patients, not bank samples. There were some bank samples. Okay, and again, these were lung transplant patients and other transplant patients. And again, we've got very good concordance between galactomannan, with a cutoff of 1, and BAL, and growth of the fungus from the BAL fluids. Okay, so we've got various different species there: Fumigatus niger, nigellans, flavus, and terreus. So it's picking up all of the different species. Again, if you look at the negative predictive value, We've got 100% for 2 of the sites, but interestingly, we've got sensitivity in 1 of the sites down at 50%. Okay, I haven't yet got to the bottom of why that's the case. Okay, but overall, the 3 centres, if you put the data together, an MPV of around 97%. Okay, so what's coming out of all of this is in the lateral flow device, it's the negative predictive value that's important for this test, not the positive predictive value. Okay, what we've also done is very recently we've looked at a whole load of patients with underlying respiratory diseases, COPD, all sorts of other breathing difficulties, AIDS with pneumonia, tuberculosis, etc. And again, using BAL fluids, comparing various different techniques, culture, PCR, wasn't PCR in this case, galactomannan, and the lateral flow device. And again, if you look, we've got a negative predictive value of about 97% for the lateral flow device when compared to the galactomannan at 1 of 99%. Okay, so we're confident, certainly for bowel, that it's a useful technique for detecting invasive pulmonary aspergillosis. In terms of serum, it's a little bit more complicated. So with bowel, there's no pre-treatment required. You simply put on the bowel fluid directly onto the device. There's no need to pre-treat it. With serum, you have to heat treat it with EDTA. So you boil it stick it in a boiling water bath or a heating block for 3 minutes with EDTA buffer. You then spin out the rubbish and then apply the clear supernatant to the lateral flow device, and again read at 5 minutes up to 10 minutes. Okay? So reasonably straightforward still, but of course that's taking it away from point of care back into the laboratory. So in terms of serum, really the most comprehensive trial we've done is with Rosemary and Lewis White in Cardiff, comparing galactomannan PCR and the lateral flow device in 103 haematology patients, 8 proven IPA, the rest probable, possible, or no infection. And in terms of the proven probable versus no IPA populations, the specificity of the LFD, 98%, similar to PCR, but better than the galactomannan test. Sensitivity inferior to PCR, no great surprise there, I don't think. And again, slightly better than the galactomannan immunoassay. So basically, in summary, the assay picks up all Aspergillus species, specific for Aspergillus. It detects activity only, the active growth phase. It's rapid, reading at 5 minutes and up to a maximum of 10. It's a single-use test, okay? And we've shown that it's compatible both with serum and BAL fluid with good results. So the test will be CE marked in May. It will come in a box with 20 devices, buffer to do the tests, serum testing, and an instruction for use in various different languages. When it comes out, in fact, ISCA will be coming off and the OLM logo will be coming onto everything, okay? The great thing is you don't have to put any of it in the fridge. It just sits on the shelf, okay? Even the buffer, you don't have to put it in the fridge. So it's really quite useful for countries that don't have immediate access to electricity. For example, we've been testing it in Africa in TB patients. Okay, so that's where we are. As I say, it'll be available and CE marked for use as a diagnostic for Aspergillus in May of this year. So that's all very exciting. What I think is perhaps even more exciting is some of the work that we're doing with the antibody in terms of actually using it as a tracer to light up infections within the lung. So I'm involved in a 5-year European project with German and French and Swiss partners. And what we're actually doing is my job has been to re-engineer the antibodies, so make single-chain Fe fragments, Fab fragments, and a fully humanized version of the antibody where we've grafted in the CDRs into a human framework, IgG1 molecule, and we've labeled it with copper-64. So this is the only image I'm allowed to show because the rest of it is all internal data for the project, sadly, but I would have liked to have shown you some more data. But on the left-hand side there, we have a mouse that's been, it's uninfected, unchallenged with Aspergillus. It's been put into MRI scanner, PET MRI, and you can see that we've got accumulation of the tracer, copper-64 label JF5 antibody in the liver. But on the right-hand side, where we've got Aspergillus infection, you can see it lighting up the lung tissue. So the whole point about re-engineering it is to make it not accumulate within the liver. Okay. And we've also, in the 5th year, we're doing human clinical trials in Germany using patients with invasive pulmonary aspergillosis. Okay, so it's costing a lot of money. It's going to cost £1 million to get the GMP-produced antibody for use in humans. But luckily, German company has just stepped in and said that they will match funds the 500,000 we got through the European consortium. Okay, so which is fantastic. So they're going to come in and not give us the money, they're going to do the work for us free of charge, which is absolutely fantastic. So we will actually get to the human clinical trial stage in year 5 of the project. And really, I think this is really the way forward in terms of diagnostics, is actually using label tracers, specific antibodies that will go to the site of infection and light the thing up. And also therapeutics, so putting on a toxin or a radiometal that's going to kill the pathogen actually in situ, which is not unrealistic. It's used for cancers, for example. Okay, so that's really it. I better stop, I think. Okay, thank you very much.